Okay guys, today's video lecture topic is going to be the classification of living things. Please make sure you are filling in your notes organizer as I go through the video lecture. So before we can start talking about how scientists classify living organisms, let's talk about why. There is an entire science dedicated to this. It is called taxonomy. So it's the branch of biology that names and organizes organisms according to their characteristics. So why? Why do we do this? By classifying organisms, we can answer questions such as how many known species are there? What are the defining characteristics of each species? And what are the relationships between those species? This idea of classifying organisms has been around for a while. The first person to do this was a Greek philosopher named Aristotle. He did it very simply. He classified organisms as either plants or animals. Well, we know that that's a serious problem because not all organisms fit as either plants or animals. So some of the limitations of early classification were that it was impossible to classify all of the organisms. What about fungi? What about protists? What about bacteria? And then, of course, they don't take into evolutionary relationships into account. So along comes Carolus Linnaeus in the 1700s, and he developed a new classification system that we still use today. His classification system was based on morphology, which is an organism's form and structure. And he basically had two major contributions. He grouped organisms in a hierarchy of 7 different levels, and he created a scientific naming system called binomial nomenclature, both of which we still use today. So we're going to start by talking about his contribution of binomial nomenclature. So why in the world do we do this? Why do we give organisms scientific names instead of just using their common names? Well, first of all, common names can be misleading. For example, a jellyfish is not actually a fish, and a seahorse is not actually a horse, but it is actually a fish. And then, of course, common names vary by location. So even within the United States, this animal right here, some people will call a puma, mountain lion, cougar, all talking about the same thing, different names. And then, of course, common names vary by language. I would call this little guy right here a chipmunk. Someone in Germany might call it a Streifenhorken. Someone in Italy would call it a tamia. Someone in Spain might call it an ardilla. So we had this sort of solution early on to fix this common name problem by using the scientific language of Latin, this unchanging, very old language. And scientists would basically take an organism and describe it using Latin. So like this red oak tree was, and I'm going to say this horribly wrong, but Quercus folius obtus cineatus cetaceo mucronatus, which meant oak with leaves with deep blunt lobes bearing hair-like bristles. So what's the problem with that? Why did we change to this two-name naming system? Well, that way was way too long, and it still didn't show evolutionary relationships. So Linnaeus comes up with binomial nomenclature, by two, nomial referencing names, so two-name naming system. The first name is the genus, the second name is the species. There are some rules to writing a scientific name. If it is printed, you'll often see a scientific name in italics. If it is written by hand, you'll see it underlined. Both of those methods are perfectly acceptable. When you write a scientific name, you always capitalize the first word, which is the genus. So capitalize the genus, leave everything else lowercase. So the species name should be lowercase, only the first letter of the genus name should be capitalized. So here are some common names. The vampire bat, scientific name Desmodus rotundus. You'll notice that it's in italics, first letter capitalized. Here's our little chipmunk again. Remember, not the Streifenhorken, I wouldn't know what you were talking about, but if you told me the Tamias striatus, then I could look it up and see that that is indeed a chipmunk. Notice that it's underlined, first letter capitalized. And then humans, you've probably heard this before. We are Homo sapiens. Okay, on a sheet of paper, I want you to answer this question for extra credit. Carolus Linnaeus was not actually born Carolus Linnaeus. He was born Carl Linné. So why did he change his name to Carolus Linnaeus? Keep in mind Latin, binomial nomenclature, think about that. Okay, so we talked about binomial nomenclature, which was Linnaeus' first contribution. His next contribution was developing these levels of taxonomic hierarchy. And remember, he came up with a 7-level classification system, starting with kingdom. We sort of added on domain, but a lot of times you'll still hear it starting with kingdom. But domain is going to be your largest category. It contains the most number of organisms. And then the species is going to be very specific. It's only going to contain one or a few organisms. So as you go from level to level, you're getting smaller and smaller, or more specific and more specific. So domain, kingdom, phylum, class, order, family, genus, species. And you'll probably want to come up with some sort of way for you to remember these taxonomic levels of hierarchy. So I remember I learned in high school, did King Philip come over for great spaghetti? And I still remember that today. You could do kids prefer cheese over fried green spinach, which I think we all know the answer to. If you come up with one on your own, it will probably be something that you would remember better, but you can always just use one of mine. So here's just showing you that a kingdom is obviously bigger and more inclusive than a species is. So kingdom Animalia contains all these organisms. Species Ursus arctos only has the grizzly bear. Again, showing you the same thing. Here's our lion. He belongs to kingdom Animalia, phylum Chordata, class Mammalia, order Carnivora, family Felidae, genus Panthera, and then species Leo. And now we're going to talk about domain. So he belongs to the domain Eukarya. There are 3 domains: domain Bacteria, domain Archaea, and domain Eukarya. Domain Bacteria has 1 kingdom, that's kingdom Eubacteria. Domain Archaea has 1 kingdom, that's kingdom Archaebacteria. And then Eukarya has 4 kingdoms: Protista, Plantae, Fungi, and Animalia. 3 domains, 6 kingdoms. So there's our 6 kingdom system. Obviously we know more now than Linnaeus did it. So very quickly, I'm just going to go through the major characteristics of each kingdom. There's a chart at the end. You might want to wait until that to fill out your chart under number 7. We're going to get into this much more later. I'm just going to give you the major characteristics of each kingdom. So kingdom Archaebacteria. Archae implies archaic, old. This is the most ancient of Earth's life forms. They are unicellular, of course. They are prokaryotic, of course. Some are autotrophic, some are heterotrophic. A major characteristic is that they live in extreme environments. Thermophiles live where it's very hot, like this hot spring here. Acidophiles live where it's very acidic. Halophiles live where it's very salty. Kingdom Eubacteria, this is probably what you think of when you think of bacteria. This contains like E. coli and staph. They are, of course, unicellular. They are prokaryotic. Some are autotrophic, some are heterotrophic. They're found in soil, water, the body. But unlike Archaebacteria, Eubacteria require oxygen. Okay, our next kingdom, Kingdom Protista, is probably one you're not so familiar with. This one has things like amoebas and slime molds, like you see in the picture, Euglena. These are eukaryotes. Remember, there's only one prokaryotic organism, and that is bacteria. So starting with Kingdom Protista, we're talking about eukaryotes, organisms with a nucleus. This kingdom is sometimes called the junk drawer kingdom because you find a little bit of everything here. Some protists are unicellular, some protists are multicellular, some protists are autotrophic, some are heterotrophic. Okay, next kingdom is kingdom fungi. These are, of course, eukaryotes again. Most fungi are multicellular. You do have some species that are unicellular, like yeast. Fungi are heterotrophic. Do not break my heart into a million pieces and tell me that they are autotrophic. They do not photosynthesize, they decompose. So they are heterotrophic. They reproduce by spores, and their cell walls are going to be made of chitin. Next up, we have our plants, which again are eukaryotic. All plants are multicellular. They are autotrophic. They make their food through photosynthesis, and they have cell walls made of cellulose. And then last up, we have the animals, the most complex of organisms. They are all eukaryotic. They are all multicellular. All animals are heterotrophic, and at some point in their life, they are all able to move, even if it's only during a certain stage of their life cycle. They do not have cell walls. They only have cell membranes covering their cells. So there's a chart if you want to pause on that and finish filling in number 7. So thanks to Charles Darwin, we actually have a lot more information on how to classify organisms than we did in the time of Carolus Linnaeus, because now we can organize living things in the context of evolution. So how can we do that? What kind of information can we use in order to classify organisms based on evolution? There are 3 major categories. Morphology, and remember I already told you was the form and structure of organisms. Biological macromolecules, which we talked about a long time ago when we talked about biochemistry. And then the fossil record. So morphology, again, is the classification of organisms based on inherited features that differ between the species. This could be looking at their bone structures, the similarities and differences in bone structures. The more similar the bone structures, the more closely related the organisms would be. Okay, you can also look at macromolecules. This is comparing things such as proteins or DNA. Organisms with similar macromolecule sequences are going to be considered more closely related. So look at this diagram here. This is looking at the amino acid differences in the protein hemoglobin. So between humans and this primate here, there are only 8 differences. Between humans and the dog, there are 32. Between humans and the bird, there are 45. And between humans and the frog, there are 67. So we would say that humans are more closely related to primates than they are to frogs based on the fact that there are less differences. And then our last piece of evidence is going to be the fossil record, where we can see changes in organisms that can be traced just by looking at the remains that they leave behind. So if you find fossils in similar sedimentary layers, you're going to consider those to be more closely related than if they were in very different layers far apart. Okay, so we can take that evolutionary history that we sort of determine based on those 3 things we just talked about. That's called phylogeny, determining evolutionary history, and we can sort of illustrate that in a tool called a cladogram. So cladistics, here's number 9 on your notes organizer. Cladistics shows evolutionary relationships based on shared derived characteristics. And then we can show those relationships through a tool called a cladogram. So looking at this cladogram here, each of these are derived characteristics. So we see that from this point on, all of those organisms have amniotic eggs. From this point on, all of those organisms have openings in their skulls for muscles because they're derived characteristics, they're shared. From this point on, all of the organisms have hair. Do you see where I'm going here? And then just by looking at a cladogram, you can tell lots of information about evolutionary relationships. We can tell that crocodiles are more closely related to birds and dinosaurs than they are to marsupials, like the kangaroo. We can tell that amphibians and turtles are fairly closely related. We can tell that marsupials and placental mammals are closely related. So if you had to guess, would you say amphibians are more closely related to crocodiles or to monotremes? Okay, so here's that cladogram again. We can tell a lot of information just by looking at how we've sort of illustrated our evolutionary relationships. Here's the one on your notes organizer under number 9C. So fill in the derived characteristics here. Vertebrae is the characteristic that all of these organisms share, so we would call it an ancestral characteristic. The rest are going to be derived characteristics. So ancestral is the oldest one, the one that they all share, and then as you go through time, you have the derived characteristics that sort of separate these organisms from one another. So can you tell me, just from looking at this cladogram, what primates are most closely related to? Rodents and rabbits. Can you tell me the characteristic that separates crocodiles and birds from primates and rodents and rabbits? Eggs with shells. Can you tell me the characteristic that separates ray-finned fish from amphibians? That would be 4 limbs. The last topic for today are going to be dichotomous keys. You probably remember these from 7th grade. This is a scientific tool that allows a user to look at an unknown organism or item and identify it based on its characteristics. So dichotomous literally means divided in 2 parts in Greek. And basically you have at each step 2 choices that you sort of follow through in order to identify an object or an organism. So here are some dichotomous keys rules. Use characteristics that remain constant. Don't use the color of a leaf if that changes through the seasons. Use measurements rather than terms like large or small. Don't be subjective. Make the choice a positive one, like something is instead of is not. If possible, start both choices of a pair with the same word or set of words. And then finally, finish the dichotomous key with the item's identity, like especially if it's an organism, finish it with its scientific name. If done correctly, you should have 1 less step than the number of organisms. So like this one, we've got 2, 4, 6, 8, 10 organisms to identify, 9 steps. So take a look at that for a second. See if you can identify maybe what the identities of some of these fish are. And then see if you can create your own dichotomous key that would identify these 4 shapes. 4 organisms or 4 objects, you should have 3 steps. And that's it for today. Y'all have a great day.